Hello, this is CJ and this is the Knowledge Catalog. We are going to tackle an important topic in biological science in this video, and if you are a public school science teacher in the Philippines, you may use this video as an advanced video lesson or as an enrichment of your discussion with your students, especially now that we are in the time of pandemic. Specifically, this video is going to clarify things in Science 8, the fourth learning competency for biological science in Science 8. And it reads as comparing mitosis and meiosis and their role in the cell division cycle. So yeah, let's begin. Cells need to coordinate the timing, the rate, and the type of cell division to conduct. If a cell is supposed to divide every other 30 minutes or every other 1 hour, then it's supposed to have that approximated number of amount of time to divide. Also, it has to have a specific type of cell division to conduct, kasi there are two types of cell division, which are mitosis and meiosis. So if the cell is a somatic cell, it's supposed to conduct mitosis, and if the cell is a gonadal or a sex cell, it is supposed to undergo meiosis. Now, if you're not familiar yet with the kind of with the terms I am talking about, I have a separate video that is discussing just that, the stages of cell cycle. Okay, so I will be giving you a link. So, in terms of the frequency of cell division, the length of the cell cycle is highly variable, even within the cells of a single organism. Different cells are going to have different lengths of cell cycle. So skin cells, they have a different length of time. They are taking a different amount of time compared to the other cells of the body. Okay, so an entire human lifetime is spent in G0 by some specialized cells, such as the cortical neurons or some cardiac muscle cells. So from birth until an organism dies, their cells, specifically their cortical neurons, are not going to divide. Okay, they are going to stay like that inside, or siguro magde-degenerate lang sila, but they are not going to divide. Okay, they'll stay in a G0 state. There is also variation in the time that a cell spends in each phase of the cell cycle. The timing of events in the cell cycle is controlled by mechanisms that are both internal and external to the cell. The mechanisms are facilitated by the cell, but the signals for those mechanisms to start may come internally or externally. Okay. The control of cell cycle happens in four different phases in the cycle. So first is in the G1 phase, which is part of the interphase, and then in the S phase, and then the G2 phase, and the M phase. Okay, I'm sorry, but I was actually I'm about to talk about the different stages of the cell cycle. So generally, it has interphase and M phase. After mitosis or M phase, some cells enter the G1 phase of a new cycle, while others will diverge at the start of G1 into a phase called gap 0 or quiescent and not dividing. This may be permanent or it may be temporary. So what happens now if halimbawa yung cell napunta siya sa G0? Pwede siyang magstay ng ganon, such as in the case of cortical neurons, like forever, kung may forever man. At pwede rin na mag-stay lang siya doon for a cell can stay in that particular state, in G0 state, and then it can begin doing cell cycle again kapag ready na siya ulit. So but yeah, cortical neurons are really going to go to gap 0. So what I'm trying to say is merong mga cells na pwede naman actually mag-skip ng gap 0, but they are going to go to gap 0 simply because they are not prepared yet for gap 1. After cell division, the daughter cells will follow one of the several pathways laid for them. So first, cells can divide again immediately enter the G1 phase of a new cell cycle. Kapag ready na siya, kahit kakatapos lang niya from cytokinesis, pwede na siya ulit mag-undergo G1 phase, which is yung preparation na for the cell cycle or cell division. And then, yung ibang cells, they can go G0 phase. Some of them are quiescent for a time, but they can re-enter the G1 phase kapag ready na sila. And some of them naman, kapag naging cell na talaga sila, daughter cells na sila, pwedeng hindi na sila divide. So yeah, in the case of cortical neurons again. And then next one is some specialized cells. For example, nerve cells do not divide again. So sabi ko nga, speaking of the devel, yan. So mitosis takes 5% of the cycling time, whereas interphase, yung all of the preparation of the cells, ayan sila, yung part na yun, it gets 95% of the cycling time. So mas matagal yung interphase, yung preparations, kesa sa mismong cell division. Next one is other cells can be triggered by activities such as wound repair to enter G1 phase of the cell cycle to divide as required. Kapag halimbawa nag-undergo ng necrosis yung ibang mga malalapit na cells, magkakaroon ng signal for mitosis. So mangyayari doon is mag-enter ng G1 phase ang mga cells near that particular wound para mag-heal nga yung wound na yun. The signaling is then because of the necrosis of the nearby cells. Okay, so the process of cell cycle are the following. Yeah, you can see the process naman, diba? So yeah, I keep on forgetting that we're just supposed to talk about the regulation of a cell cycle. So yeah, the process of cell cycle. Chemicals called mitogens are going to signal the start of cell division. This is, if I'm not mistaken, it can be autocrine or paracrine. If I'm not mistaken, the signal can come to the cell autocrinely or paracrinely. Now G1 takes almost half of the total cycling time, such as seen here, almost half of it. And it can lengthen depending on microenvironmental conditions and signals received from other cells. So during this step, there is one DNA copy only. Okay, during gap 1, there's only one DNA copy. Licensing system that expires by the start of S phase. Again, there is a one DNA copy only licensing system that expires by the start of S phase. Why is that so? We all know that S stands for synthesis. And if halimbawa for gap 1 allowable na dapat meron lang siyang isang DNA copy, then it means mag-end na yung licensing system na yon upon entering the synthesis phase. So how is that going to be checked? It is going to be checked by the G1 checkpoint. So again, in G1 checkpoint, i-check nila kung na dapat meron lang isang DNA copy galing sa G1. So at the molecular level, a cell cycle checkpoint consists of a sensor or detector, a signal center, and a receiver or effector. So G1 checkpoint checks for DNA integrity. Kailangan yung DNA na meron ng cell is okay siya, or damage usually by the tumor suppressor gene P53 of chromosome number 17. Chine-check again ng G1 checkpoint kung yung DNA integrity, at nag-check din siya for damage. Usually, yung damage lang naman ng cell is yung tumor suppressor gene P53. Kung active ba siya, kung okay ba siya. If the DNA is damaged, repairs are done. May slow down and yung process, and then ma-arrest yung cell cycle, hindi siya magpa-proceed. But if the damage is beyond repair, P53 gene will signal apoptosis. So P53 is basically the guardian of the genome. Siya yung papatay sa mismong cell. Again, kasi apoptosis is programmed cell death. yung p53 gene ng mismong cell na yon is going to signal apoptosis kasi nga beyond repair na yung damage niya. So, hindi pwedeng kumalat yon sa mga daughter cells. Kung sakaling magpo-proceed ito, kakalat yun. Siyempre, from only one cell, magiging dalawa na silang merong erroneous na DNA. Yeah. So, before we proceed, I hope that you check that p53 is going to signal apoptosis if the DNA damage is beyond repair. Okay? P53 can be damaged, okay, by itself. Yung mismong guardian, pwede siya ma-damage by UV radiation and mutagenic materials or chemicals from cigarette smoke or aflatoxin from moldy peanuts, for instance. May mga ganon. Yung mismong guardian ng cell division or yung mismong guardian actually ng genome. Yung mismong guardian ng genome, pwedeng siya mismo yung ma-damage. So we have to make sure that we are keeping ourselves safe from these factors that can damage p53 gene. I hope I'm making sense. In the G1 checkpoint, we are checking for DNA integrity or damage. Ayan. So, that damage is going to be checked by the tumor suppressor gene, p53. Okay. Next is S phase. Sa S phase naman, only one new copy of the cell's DNA is synthesized. The double-stranded DNA unwinds into two component strands that serve as templates for the synthesis of a new strand on each. We are not going to dive into the details yet because I'm expecting that if you are going to teach grade 8 students, they may not need to know yet kung ano yung specifics nito. S checkpoint checks for shortage of DNA nucleotides or presence of some enzyme inhibitors. So, kailangan the DNA is completely replicated, or the new DNA is completely synthesized at this stage. Okay, next one is G2. So, G2 is shorter than G1 as seen here, and this time is spent in developing and preparing organelles for dividing and sharing during cytokinesis at the end of mitosis. So, dapat at this particular time, yung mga organelles na dapat na duplicate na sila. And dapat yung cell is already large enough for it to start dividing. Now, G2 checkpoint. It checks for the DNA damage kung meron pa rin. Okay? Siyempre, dalawa na yung DNA copies dito, diba? Okay, so, I mean, there are two strands of DNA kasi yung isa original, isa naman yung, basta, there are two pieces. Na-copy na kasi siya, na-synthesize na siguro. That is that's a proper way of saying it. Na-synthesize na yung isa pang DNA or nakapag-synthesize na ng DNA sa synthesis phase. Ang gulo ko, no? Okay, so dapat yung DNA na yon, yung dalawang DNA na yon, walang damage. Okay, so kapag merong damage, it's in G2, mare-repair yun. Ngayon, kapag beyond repair na, i-reject siya. Now, if there is an unreplicated DNA and checks for mechanisms leading to separation of sister chromatids, kung meron... Okay, so let's discuss this again. Ganito. In G1 phase, there is this one DNA copy licensing system that will end by synthesis phase because a new DNA strand is going to be synthesized. Now, upon reaching G2 phase, that DNA, those two DNA strands must have integrity. And if they are damaged, in G2 checkpoint mismo, madedetect yun, and they will be repaired, or they can be rejected. Ngayon, kung meron pang unreplicated DNA, syempre hindi naman pwede mag-proceed ng mitosis kapag ganon. So, mahal din yung process. And then, if okay na meron ng dalawang DNA talaga dyan, wala namang ibang problema, kailangan i-check pa rin yung mechanisms para mag-separate yung dalawang sister chromatids from each of them. So, that is still going to be checked by G2 checkpoint. Now, let's proceed. At mitosis, the sister chromatids joined as pairs by some cohesive glue. Since they were replicated in S phase, are separated to form two new equivalent sets of chromosomes. So this event is called chromosome segregation. In metaphase checkpoint naman, which is also known as spindle assembly or kinetochore attachment checkpoint. So how many checkpoints do we have so far? One checkpoint. We have two checkpoints, and then the third checkpoint is right here. It is called the M checkpoint or metaphase checkpoint. Okay, let us be particular with that. It's not mitosis checkpoint, it's metaphase checkpoint. It checks for some misaligned chromosomes and that microtubules are attached to the kinetochores, a very critical mechanism. The start of anaphase is delayed until all of the chromosomes are aligned and they are appropriately attached. So if you have seen one of my if you have seen my other video that describes this process, I hope that you remember that the microtubules from the centrosomes, they can actually have two roles or two actions. Okay, for some microtubules, they have to connect to the kinetochore. So that is like, kinetochore is a protein na nakasurround or malapit sa centromere. Kailangan na-connect ang microtubule sa mga kinetochore na yun. And the other kinetochores naman must reach for the other kinetochores from the opposite pole so that they can push each other away. Push each other away. Why is that so? Because kailangan magkaroon ng mechanism para mas mapa-elongate pa yung cell at mas mapadali yung pag-form ng two new daughter cells. Kapag hindi pa aligned yung mga kinetochore, yung mga microtubules, pag hindi pa sila nakakonect sa mga kinetochores, hindi magi-start ang anaphase. Kapag hindi pa stable yung mga free na mga microtubules, hindi rin magi-start ang anaphase. So unless those two conditions are met in the metaphase checkpoint, anaphase is not going to take place. Actually, it can't take place. Okay. So the activation of cell division. Activation of cell division is regulated by chemical signals in the cytoplasm, which are usually proteins that can act as activators or inhibitors. So when we say activators, these are chemical signals that start a process, whereas an inhibitor naman, these are signals that will stop a particular process. So external signals such as the death of a nearby cell or the release of hormones such as human growth hormone or HGH can trigger cell division. However, too much HGH may result to gigantism, which is the opposite of dwarfism. So discussing this next text box is that first we need to talk about death of a nearby cell. So we mentioned kanina necrosis. Kapag nagkasugat ang isang tao in a particular part of their body, yung mga cells na doon, they died. So yung mga nearby cells na yan are going to do cell division because they can use the death of the nearby cells as their signal. However, if wala namang sugat, pwede rin naman yung pagkaka-release ng human growth hormone or ng mga growth factors. Kasi yung mga yun, mitogen din sila. Or they are signals that can start, what do you call that? Mitosis or, somatic cell division. Others naman, other growth hormones include platelet-derived growth factor or PDGH, which stimulates cell division in the fibroblast or connective tissue to heal wounds. Now, PDGH or, I'm sorry, nag-repeat lang yung text box na yan. Okay. So, cancer. Cancer can get to be caused by switched on proto-oncogenes or normal growth factors that mutated, say for instance, RAS or rat sarcoma. Okay, or switched off tumor suppressor genes or P53. So, what does this mean? we all have proto-oncogenes in our genomes. May mga may mga rat sarcoma tayo, meron tayong RAS. It's a super family of G proteins. Now, these, when they're turned on, it can cause cancer. That will cause cancer. And then, kapag na-turn on na to at naka-off din, actually, ang ko dito is, ang pagkakaintindi ko dito is that pag na-switch off ang tumor suppressor genes, magta-turn on ang mga proto-oncogenes. Pero, if they are separate events, then it is going to worsen if even these tumor suppressor genes, P53, the guardian of the genome, is switched off. Because, it's a gene. It can be turned on, it can be turned off as well. So, cancer develops, ultimately, if the following conditions are present. So, first is yung mga growth genes are turned on. Tapos, they stayed like that. Second one is, growth-promoting genes stayed on because, tumor suppressor genes such as P53 were turned off. So, yeah, B, na-ignore yung checkpoints. C naman, na-escape yung apoptosis. So P53, since it cannot release a signal for apoptosis, then naka-turn off na rin yung mga suicide genes. Okay, immortality naman or unlimited divisions, naka-turn on ang chromosome maintenance genes. For angiogenesis naman, naka-turn on ang angiogenesis promoting genes. So let us clarify muna that angiogenesis means the mechanism or the production of blood vessels. That's actually good for the spread of cancer cells. Now, F naman is overcoming anchor and density dependence. So naka-turn off ang touch sensor gene. So, those cells can just roam around. Cell mutations can be triggered by ultraviolet radiation, yung sabi kanina, because UV radiation can turn off P53 or a particular tumor suppressor gene. Chemical or radiation exposure, heat, cigarette smoke, pollution, age, and genetics are all of the other factors that can trigger cell mutations. So yeah, that's right. Age is one of them. And yeah, even genetics. Totoo yun. May mga tao talaga na tinatawag nilang merong cancer sa lineage talaga nila. In their family, it runs in their blood. That's true. Now, tumors are masses of abnormal cells. Kapag halimbawa ang tumor is, it remains in the original site, lump lang siya, and then it does not really cause any serious problems. Pwedeng matanggal by surgery. It is benign. Okay, and yeah, na-halt na rin yung P53, medyo paulit-ulit tayo doon. And then malignant, cells metastasize or they start more tumors, impairing the functions of organs throughout the body. So by saying metastasize is just that the cancer cells have overcome yung sinabi na kanina yung touch sensor genes, naka-off na yun. So, they can just flow into the bloodstream. And if na-turn on din yung angiogenesis promoters, yung pag-produce nga ng blood vessels, then that's going to make the cancer more deadly. Now, we have several treatments for cancer. Treatments target rapidly dividing cells, high-energy radiation, and chemotherapy. What I've heard about the high-energy radiation, or what I learned about it, is that tina-target niya talaga sana is yung mga parts ng tumor na, if I'm not mistaken, may oxygen. Yeah. Pero yeah, that is generally what we know about it. High energy radiation and chemotherapy. Some naman are drugs targeting some proteins or enzymes found only in cancer cells, and in my reference, it's one of them is Gleevec, some drug called Gleevec. And so I know that if you just watch this video without first understanding what are these stages of cell cycle, this may actually come as a very confusing video. But yeah, if you were able to watch my other video about the cell cycle, then I hope that you had some better understanding on are those processes, yung mitosis and meiosis, maintained. How are they regulated? So remember the checkpoints. G1 checkpoint, we have G2 checkpoint, and then we have the metaphase checkpoint. Those are really important, and I guess that the most important word that you learned in this video, and I hope that you indeed learned it, it's P53, a kind of a tumor suppressor gene. The Knowledge Catalog is a brand being developed by yours truly, who is a teacher creator. I am a high school teacher in the Philippines, science, and I love developing content that I find useful, and things that I need to better my instruction. You can show your support by leaving a like on this video, by sharing this perhaps to some of your colleagues or classmates, and you may subscribe to this channel even to follow me as I create more videos like this. If you need, or if you want to have a copy of this presentation, you may contact me through my page. Yung link nasa may unang slide, yeah, but I may tell you, it's facebook.com/knowledgecatalog/. Thank you so much.